Good morning. What I'd like to do today is to continue to talk to you about what I think is probably the topic that most students find the most interesting, the topic of the plant growth regulators. The last time I introduced you to the first two which were discovered, namely the auxins and the gibberellins. And after the discovery of these two growth regulators and people's understanding of the profound effects they had on development, there was the notion or the hypothesis that there must be out there many more growth regulators, and they would have as profound effects on development as did the auxin and the cytokinins. And the question one has, if you want to start looking for other growth regulators, is you know nothing about them, you know nothing about what they're going to do. How do you begin to find these new growth regulators which you hypothesize are out there? And so what I'd like to do is to begin by showing you the system that people developed to begin to hunt for new growth regulators, but not knowing what exactly they were looking for. And this system depends upon a cell type that you are already familiar with. This is a cross-section of a eudicot stem. And the tissue which we're going to use for this discovery of new growth substances is in the very center of the stem, and it is the pith tissue. We're going to talk about how we have used this tissue for finding out new growth regulators. Now, there are a couple of characteristics that we want to note about the pith tissue. The first, the pith tissue consists of undifferentiated parenchyma cells. So this is the simplest cell type that we talked about, from which all other cells are derived. And the second characteristic of the pith cells, which made them very suitable for the hunt for new growth regulators, is that they are sterile, or as plant biologists call it, they are aseptic. That is, not contaminated by microorganisms. And this is an important characteristic, this sterility or aseptic nature, because what we wanted to do is to know what was around the cells, that we were dealing only with the parenchyma cells and not with some other microorganism that might be contaminating them. So let's talk about what was done and how people then began to examine for the new growth regulators. These aseptic, undifferentiated parenchyma cells were transferred to a flask which had on it a agar medium, an agar nutrient medium. And here are the parenchyma cells, or pith cells, onto the agar medium. So this agar medium has nutrients in it. And we call this type of experiment a tissue culture experiment. This is tissue culture. You are taking a tissue or a region from the plant and putting it into aseptic conditions, in this case, the tissue culture conditions. And now what was done was to say, okay, I would like to see if I can find some new growth regulators. How would I go about doing that? So what individuals did is they took plants, or maybe a leaf, whatever part they were interested in, and they ground it up, and they began to extract various fractions of the thing they ground up. So, for example, they may have extracted a fraction which partitioned into water, or a fraction which participated into organic solvents, or an acid fraction, or a base fraction. So they ended up with many hundreds of different fractions from the extract from the plant. And each of those fractions were added aseptically to the culture medium. And then we let some time go by, and we asked, how did the pith or parenchyma cells respond? Did they give any particular specific developmental event? And in one of the flasks, or one of the fractions that was looked at, what was found was that if we waited for some time, so we waited for some time after adding the fraction to here, the fraction was added, So here we have our medium again, our culture medium, whatever it happens to be, containing the fraction that we're looking at. What they found after some time was that the pith cells divided into a mass of undifferentiated cells. So that if you waited for some time, the original small explant of pith cells divided into a mass of undifferentiated cells. This term, mass of undifferentiated cells, plant biologists call a callus. So a callus is cells which are undifferentiated and which just divide. That's all. So these individuals then, having discovered that there was something in the medium which caused the cells to divide, began to ask what was that substance. And coming from this work was the discovery of the third group of plant growth regulators, which were known as the cytokinins. The third group of growth regulators. Now, as with the other two growth regulators, these do many things in plants. And what I want to just do right now is to give you the tip of the iceberg with regard to some of the things that are stimulated by the cytokinins. First of all, they stimulate cell division. This is the basis of their discovery. Secondly, a very interesting aspect of cytokinins is that they delay senescence. That is, delay getting old. So it got people quite interested in them. The third aspect of cytokinins is that in combination with other growth regulators, and the main growth regulator that they were put in combination with is auxin. In combination with other growth regulators, they can profoundly affect plant development. And I want to explore this statement a little more by showing you what was done. So, culture flasks were made which had varying amounts or ratios of cytokinin to auxin. And the tissue was then put into these flasks, and some period of time was allowed to pass. And I'd like to give you a summary of what was found, depending upon whether the cytokinin was high or the auxin was high in the medium. And on this axis, we'll put auxin. This is auxin concentration, and it's increasing in this direction. So 0 is down here at the bottom. And on this axis, we'll put cytokinin. And it is increasing in concentration as we go to your right. Now, when the amounts and the ratios of auxins and cytokinins were varied, what was found was that if the cytokinin level was low in the culture medium and the auxin level was high, you tended to produce roots. And if the cytokinin level was high and the auxin level was low, you tended to produce shoots. And in between, you could produce all variations, including undifferentiated. Tissue. So by altering simply the ratio or the amounts of these two particular growth regulators, it was possible to profoundly affect development. And today, This is a very puzzling system. No one understands how it is when you change the ratio at a molecular level. You're able to cause the cells to follow a pathway that results in the differentiation of the root, or cause the cells to follow a pathway that results in the differentiation of a shoot. A very profound question, and yet we really have very little understanding of it. Now, what I want to do is to talk about the fourth of the general effects of cytokinins, and then we'll look at some pictures of these. The fourth effect of cytokinins is that they release apical buds, or let's say axillary buds. Axillary buds from apical dominance. You'll recall that apical dominance is the effect where the main shoot apex produces auxin, which then inhibits the outgrowth of the axillary buds on that same stem or that same branch. So the way that experiment is done, where they can release these axillary buds from the apical dominance, that is, overcome the inhibition of auxin, is we'll have a stem. Let's just put our stem here. Here we have a inhibited axillary bud in the stem over here, like so. And what is done is to take some cytokinin, and you can do this by putting a little bit in some Vaseline or in some type of lanolin solution, and applying, we can apply cytokinin directly to the bud. And when you apply cytokinin directly to the bud with time, the bud grows out. Now, this observation that cytokinins can overcome apical dominance is the basis for understanding some plant diseases. There is a disease that some of you may have seen called witch's broom. In which, if you look at the end of the branch, all of the axillary buds have been released from apical dominance, and it looks like the broom of a witch that you would see, for example, in The Wizard of Oz. This release of the axillary buds from apical dominance is due to the fact that the pathogen that is inhabiting the tree either itself or causes the tree to make more cytokinins, and the increased cytokinin level causes these axillary buds to grow out. Additionally, there are some pathogens which cause massive cell division on trees, and these cause tumors, just like you find in people, where you have an outgrowth of lots of undifferentiated tissue to form a structure which you know of as a tumor. Let's look now at some pictures of cytokinin. Why doesn't this want to go here? Okay, let's do this here. We should get it next. Play. Okay, this first slide shows you the callus tissue forming. And if we work backwards, this callus tissue originally came from some pith tissue, which put it onto agriculture medium, and then with time, the cells divided and produced this undifferentiated mass. This is the basis for the discovery of the cytokinins. Here is a flask now in which the medium has been heavily biased towards cytokinins, and the callus tissue has regenerated a shoot system in the beaker. As contrasted where we now have a beaker or a medium which contains a higher ratio of auxin, and in this particular case, you differentiate roots. Here is that same sort of chart that I gave you before, and the important points on this chart to notice is that when the auxin concentration is high and the cytokinin concentration is low, you mostly get roots, and when the cytokinin concentration is high, you mostly get shoots. And in between, you can get undifferentiated cells or variations of the root and shoot system. Here we have now our inhibited axillary bud. This is a P stem. There's an axillary bud inhibited growing over here. And if you apply a drop of cytokinin to this axil, you can cause the bud to overcome apical dominance, to overcome the auxin, and to grow out. And here's the witch's broom. This is a branch which is growing up. This branch is now pathogenized by some bacterium, which is causing a large amount of cytokinin to be produced, and the buds grow out. And here's a tumor, again due to a pathogen which has infected the plant, causing an increase in cytokinin. And I think the last slide, for those of you who've been up to Davis and who've been along the quad, what you will find is some of the oak trees along the quad have these large outgrowths, which are tumors, and these tumors, again, are likely a result of an infection by some microorganism, which is important with regard to causing the cells to divide. So these are the cytokinins. They are an important group of growth regulators as far as development is concerned, but not too important commercially, at least not right now. So we've considered 3 growth regulators so far, the auxins, the gibberellins, and the cytokinins. Each of these growth regulators is distributed throughout the plant by the vascular system. The growth regulator now, which is the 4th one that we want to consider, unlike the previous 3, is not distributed in the vascular system. It's a gas. It's distributed in the atmosphere. And the discovery of this particular growth regulator is very interesting and came about more by accident than by design. In the late 1800s, houses began to be heated by gas, natural gas, and also to be illuminated with gas lights. And it was noticed that in the rooms of these houses, which had either gas lighting, gas heat, or both, that the plants, the potted plants people had growing in their houses, died. But before they died, they exhibited a number of symptoms. The first symptom they exhibited is that the petals on the flowers discolored and fell off, which we say they abscised. The second aspect of these is that, do you want to get it? The second aspect of these that we want to notice is that the leaves on the plant exhibited a type of development in which the leaves bent back on themselves. They curled under, like so, which is called epinasty. Epinasty. And then finally, the leaves abscised or fell off. Now I'd like to just briefly talk to you about how the leaves fell off, how they abscised, because we're going to be referring back to this in a moment. Here is a stem with a leaf. So here is the leaf. Here's the stem. And what people discovered is that when the leaves abscised, there was a zone here where the cell walls weakened. And this zone, because that's where the leaves abscised or fell off from, was known as the abscission zone. So where the leaf was attached to the plant, there was this weakening of the cell walls which occurred in these rooms that were illuminated by the light or the heater that was turned on using natural gas. And when individuals then began to examine what it was about the natural gas, there were a number of discoveries that were made. The first discovery that was made is that a few plants would grow in the room with the heat or the light. And this is one of the plants. For those of you who are Victorian fans, if you want to see an accurate representation of what a Victorian room would have looked like, this would have been the only plant that could grow. This is known as the cast iron plant, and for some reason, it does not seem to be affected by the contaminant or whatever it was in the natural gas which was being released into the room. So this plant, the cast iron plant, is the only plant you should see growing in a movie which is accurate as far as the setting of the room container. So what did people find was the contaminant of the natural gas causing these phenomena and then also causing the abscission zone to activate? What was discovered was that the natural gas contained a contaminant, a low molecular weight contaminant known as ethylene. And ethylene turns out to be our fourth growth regulator. Which in low concentration has profound effects on development. We want to talk about these effects. The first thing I want to say to you is that in addition to these particular characteristics, which we have listed that ethylene or this contaminant in the gas caused, the fourth characteristic, which was discovered and which humans have exploited, is that ethylene is produced in fruits which are ripening and apparently cause the ripening. So fruits make ethylene, and then the ethylene causes the fruits to ripen. But as importantly, with regard to this ripening process, and we're going to return to this statement in a moment, ethylene production, and I'll just abbreviate ethylene with an E, ethylene production is autocatalytic. That is, it stimulates its own production. Of the growth regulators that we will consider in this class, the one which is most important economically is ethylene. And it is most important because it is the substance which controls fruit ripening, and economically, this is an important aspect of our lives. Fruits such as apples, melons, oranges, bananas, all have their ripening controlled and stimulated by ethylene. For those of you who go to the store and find that you have a banana which is green, the bananas which are green represent a fruit which has been harvested in this particular condition in order for it to be able to transport a long distance. And prior to the banana being put out onto the shelf for you as a consumer to buy, the produce manager takes the banana and exposes it to an atmosphere of ethylene. We'll talk about that in a moment. And that process of exposure to ethylene triggers the ripening process and also causes the starch in the banana to convert to sugar. So if you're among the first to enter perhaps Monterey Market or Berkeley Bowl when the store opens, go over to the bananas, and while nobody's looking, just sort of feel them, and you will find that the bananas are warm. And the reason they're warm is because they have come from a room where they have, before the time they were put out, been treated with ethylene gas. Now, not only is ethylene gas produced by ripening fruits, and it's autocatalytic, but ethylene is also produced by damaged fruits. So if you buy a green banana at the store and it's not ripening fast enough for you, or you want to have it ripen faster, what you can do is take an apple, which is either already damaged or which you can damage by cutting, and take this damaged apple now, in which you are going to stimulate ethylene production as a result of the wound. Put it in the bag with the green banana, then take the bag and seal it, and wait for, in this case, a very short period of time, and then the banana will ripen. Okay? This Ripening occurs as a result of an accumulation of ethylene in the bag. And for those of you who have heard of the saying, one rotten apple spoils the barrel, that saying is based upon the fact that if you have a damaged apple in a barrel of good apples, that damaged apple will continue to make ethylene, and that ethylene then will cause the rest of the apples in the barrel to ripen. So now let's explore a little bit more about ethylene. First of all, there are some fruits, such as kiwis and avocados, where the fruits do not ripen on the tree. The only way you can get these fruits to ripen is by picking them, and in picking them, you damage the end where they were attached to the tree, and you stimulate ethylene production. Now, as we said, the greengrocer or the produce manager can manipulate the atmosphere surrounding the fruit in order to get the fruit to ripen. Fruits are always producing ethylene, and the question is, how do you keep the ethylene levels low enough so that you can have the fruits not ripen too quickly, but be ready at the time you want to put them out on the shelf? You could store the fruits in a room where you're constantly changing the air supply, and in that way are able to then manipulate the environment around the fruit and delay the ripening. But a discovery was made, which of course came out of the fact that understanding ethylene control of ripening is so economically important. This discovery that was made is now widely applied and used in ripening and in controlling ripening of fruits. And that discovery is that CO2 and ethylene compete for some site. We're not quite sure what it is. You could say it's a receptor. Some site which regulates ripening. Normally, CO2, as most of you know, although with global warming it's changing, normally the concentration of CO2 in the atmosphere is 0.03 to 0.04 parts per million. What has been discovered is that if you store fruit in an atmosphere, a CO2 atmosphere of 5 to 10%, containing oxygen maybe 1 to 3%, that you can delay or in many cases inhibit ripening. So a lot of fruits, once they are picked, are put into this type of an atmosphere, which even though ethylene is being produced by the fruit, it doesn't have a chance to be autocatalytic to stimulate more of its own formation because there's a competition for some receptor or some site by the CO2, a very important aspect. So ethylene is extremely important with regard to keeping and maintaining fruits and understanding when they can be made ripe and preventing spoilage. It's a really important compound. Ethylene has some other very important commercial aspects, and this has to do with promoting flowering. And in particular, two crops where flowering is promoted by applications of ethylene are mango and pineapple. These two crops are difficult to harvest. Pineapple is a very uncomfortable plant to harvest. It's very expensive to harvest pineapple. And so what is done is fields of pineapple and orchards of mangoes are treated with a compound which causes an increase in ethylene, and this stimulates flowering so that the flowers are all sort of stimulated to come out at the same time, and therefore the fruits all develop at the same time and are ready to be picked at the same time. So this is a real important aspect of ethylene. The sixth aspect of ethylene, which is also commercially important, is that it is used in harvesting certain fruits which normally involve a lot of human labor. Fruits such as cherries and walnuts, where you have to individually pick each of these fruits, take a lot of time, can make the fruit harvesting quite expensive. And so, again, what is done is to treat these orchards with a compound that causes an increase in ethylene, and the increase in ethylene yields, and this is a point that we'll refer back to, a development in the cherry and walnuts of an abscission zone. That is a weakened region where the cherry or walnut is attached to the tree. And I've given you a link to a YouTube video showing you a farm in the Central Valley of California in which the trees have been treated with a compound that causes the abscission zones to form in the trees. And then what is done, as you will see in the YouTube video, and I hope you look at it, it's quite spectacular, is a tarp is put under the tree, and a machine, looks like something from a science fiction movie, comes over and grabs the tree and shakes it. And because of the weakened abscission zone, the cherries or whatever you're trying to get off, whether they're walnuts, fall from the tree relatively easily. So this compound, ethylene, has many, many effects on our lives. It affects your life daily in the fruits and vegetables that you eat, and particularly in deciding whether or not they ripen or whether or not they spoil. This is an important growth regulator, and again, it's naturally occurring, so it can be applied to the plants, and it's not considered to be something which we humans have not been exposed to earlier in our lives. Let's now look at a few pictures of ethylene. Okay, the first slide is to show you this epinasty which develops. Here is a plant, which is the control plant, and if that plant is put into a room with high ethylene concentrations, the leaves curl back on themselves, as the Victorians discovered. Here is the cast iron plant, which I have up here in front. This is a plant which was used in Victorian times, and in the end was one of the few plants which could be grown in the Victorian rooms that were either illuminated by gaslight or heated by gas. Here are bananas. Most of you probably appreciate that bananas are picked green, and they are then shipped up to wherever they're going, and the shipping containers contain this modified atmosphere, which generally consists of a high level of CO2. Now, this is the kind of damage that some of you may have found in some apples that you purchased from the store. If the atmosphere inside the holding area is not regulated properly, the high levels of CO2 can actually cause damage to the apples, so that you have to be able to very precisely regulate this atmosphere in order to assure you're not damaging the fruit. Okay, so this is ethylene, an important growth regulator, and as I said, the most important one with regard to commercial applications. Finally, what I'd like to discuss with you is the last of the growth regulators that we'll be able to talk about in this class, the fifth group of growth regulators. Now, this growth regulator was discovered in the 60s and the 70s, 1960s and 1970s, simultaneously by two different groups. One group working in Wales, in the United Kingdom, and the other group working at UC Davis. Let's talk first about the group at Wales, how they went about to make this discovery. They worked with shoots, which all of you can see outside. If you go outside now, you'll find dormant shoots on plants. And these dormant shoots, if you look at the very end of the dormant shoot, they have the buds that are going to come forward. So here's the shoot. Here's the bud on the end. But this is a dormant bud. Presumably, it allows overwintering. Allows overwintering. And if you look carefully at the bud, as they did, there are scale leaves. Scale leaves which cover the outside of the bud. Now, what these workers did was to remove the scale leaves and what happened is the bud grew out like this branch over here, the same plant. The leaves grew out of the plant. So if you remove the scale leaves, the dormant bud now activates. So the dormant bud, or the dormant shoot, activates. And so the workers who were studying this system hypothesized that there was something being produced in the scale leaves which was enforcing dormancy, which was keeping the shoot system from growing out. And coming from this work, the group in Wales identified, again, using various sorts of systems, a biochemical or a compound which they called dormin, because it promoted dormancy. At about the same time this group in Wales was working on this compound called dormin, there was a group in Davis who were approached by a large number of farmers here in California. And these farmers were mostly cotton farmers. And the cotton farmers had the following problem. When the cotton fruits were developing, and again, to remind you, this whole structure is a fruit. The seeds are on the inside, surrounded by the hairs that we said are the cotton hairs. Presumably the hairs are important for the dispersal of the seeds. But what the farmers came to the people at Davis for was the fact that in many of the cotton fruits, in the month of June, we found that there was a dropping or an abscission of the cotton fruits, so-called June drop, they described it as. And of course, when the cotton fruits fell off, that decreased how much money they could get because they could harvest less cotton. And so they asked the people at Davis to try and analyze what was going on in these plants. They didn't have a disease. That was quite clear. They didn't have a disease. But there was something else going on in the plants which caused this June drop and hence a loss in their money. And so what the people at Davis did was to, maybe we'll go over here, to take, just as we saw in the very beginning with the discovery of cytokinins, to take cotton plants at various stages of their development and to analyze what chemicals were being produced by the plant. So if we have an axis here, and on this axis we have the June drop, and this is increasing, and here we have time. What would happen is that maybe in March, April, and then June, we would have very little going on, and then by the time we got to June, it was increasing. So these are the cotton fruits falling off. Excising. And what they discovered was there was a chemical, one of the many substances, which also followed the same pattern and increased just before the June drop occurred. A substance which increased just before the June drop occurred. And this substance was identified chemically, and it was called abscisic acid because it promoted abscission. And it is now known that abscisic acid and dormin are the same. They're the same substance. But by convention, this compound is now called abscisic acid and not dormin, and it is abbreviated as ABA. Naturally occurring growth substance. It also has profound effects on plant development, again, in many different concentrations, but primarily the effects on development are at low concentrations. And I'd like to discuss with you now some of the effects that abscisic acid has on development and how important it is, especially in some unusual ecological situations, for the survival of the plant. So the very first thing we could say about abscisic acid is that it is a compound which is mainly, but not completely, associated with stressful conditions. So ABA with plant stress. And I'd like to give you a couple of examples of the stressful conditions in which ABA is associated. The first condition will involve a plant growing in the soil where we have drought occurring. Drought occurs. If there's a drought occurring, that means that there's less and less water in the soil. So the plant is continuing to loss of water via the stomata. We're going to talk more about this later on. Now, what happens in a drought situation? Well, it's more and more difficult for the plant to get water, and yet it's still losing water. This plant potentially could die. It could dry up because it continues to lose water, but it can't replace it from the soil. So in drought conditions, what happens is that the plant roots sense the drought. They make ABA. The ABA moves to the leaves and causes the stomata to close. So although the plant does not have a nervous system, it has a way of communicating conditions at one end of the plant to the entire plant system. Another stress condition which plants are now increasingly encountering is that the soils that are used for cultivation are increasingly becoming more salty, either as a result of evaporation from the soils or as a result of the intrusion of seawater into agricultural lands. Plants have a difficult time in saline environments. A salty environment is also known as a saline environment. And what is found is that in this circumstance, ABA is synthesized as before in the root and causes or leads to the production of proteins which help the plant deal with salt stress. Finally, what I'd like to do is to talk to you about what I think is the most interesting developmental aspect controlled by ABA, and that has to do with seed physiology. As most of you know, seeds are produced in the fall, and then you plant them in the spring when the conditions of the weather get more favorable for seed germination. But in natural environments, these seeds, which fall to the ground, have to know when it is time for them to germinate. What is it that tells them that the environmental conditions outside now are favorable for the seed to germinate? And in these circumstances, what one has found is that in the seed coat, when the seed is made, there are high levels of ABA and some other inhibitors. And these high levels of ABA and other inhibitors prevent the seed from germinating, just like the scales prevented the shoot from coming out. And now, this is the really interesting aspect of it. How does the seed know when it's time to germinate? It's got that ABA and the other inhibitors in the seed coat. How does the seed know when to germinate? And the answer is that ABA and the other inhibitors, but I'll just write ABA, ABA is degraded by the cold. That it is cold labile. So what happens in a natural environment is that the seed falls to the ground, winter comes along, and now there's a period of cold. And during the period of time that there's cold, the ABA is gradually being degraded, gradually disappearing, so that eventually it reaches a level in which it is no longer inhibitory, and presumably when it reaches that level, it's a proper time to germinate in the environment. So ABA, in a sense, is the way the plant measures the environment and decides whether it is the right time to germinate. The last aspect of ABA, and this effect of cold on seed germination is used commercially for many different seeds. Some of you may actually buy some seeds to plant in the springtime, and on the seed it'll say, on the packet it'll say, put this in the refrigerator for a month, put it in the freezer for a month. What you're doing is you are essentially causing the breakdown of the turnover of the ABA. And this process of breakdown of the ABA or the other inhibitors with cold is called stratification. And is of widespread commercial interest. Finally, I just want to point out to you one other interesting seed-related control which is attributed to ABA. In the desert, the big problem for a seed to get going is whether or not there's going to be enough water. So a seed is made, falls to the ground. How is that seed going to know that there's adequate water to be measured, to allow growth to occur? So again, in the desert, we have a seed coat which contains ABA and perhaps some other inhibitors. Now, this seed has to know that there is an adequate rain which will allow the seeds to germinate. And so the way this seed determines whether or not it's going to germinate is if there is an adequate rain, and that is a big enough rain to support the seed to establish itself, the seedling to establish itself, that rain is also big enough to wash out the inhibitor from the seed coat. So a rain that is adequate enough to support the seedling development is adequate to wash out the inhibitor, and that's the signal to germinate. Now, this system can be tricked. You can have a lot of short rains, which wash out a little of inhibitor with each rain, and then the seed would germinate, and perhaps the conditions wouldn't be very good. But in general, this is a terrific system for measuring the amount of rainfall, that is, correlating it with the amount of inhibitor which is washed out. Let's look now at a few pictures. Here is our cotton, and this cotton, again, represents the plant in which abscisic acid was first discovered, and it was purified, and again, where we had the June drop by the farmers. Here we have the dormant buds, and again, I want to draw your attention to the scale leaves, which were the site of the compound that was called dormant. And here is the germination of a seed, and in seed germination, one has the removal of both ABA and other inhibitors. Finally, I want to mention to you that there are some seeds in which there is no dormancy at all. The seeds actually germinate right on the plant. And those of you who have opened watermelons up or perhaps ears of corn up and have seen seeds germinated on the cob, or you've seen seeds germinating in the watermelon, they germinate because they don't have any inhibition to germinate. Probably they have a defect in their inhibitor content. This is mangrove. In mangrove swamps, for those of you from southern Florida, where the seeds are actually germinating from the fruits while still attached to the plant. And here you can see the series of events. Here is the flower, and this is the fruit over here. And as the fruit then matures, it germinates to produce this new shoot system. Finally, what I want to just take you back to is a point that I made in some of the labs yesterday. I want to give you some practical information. And that practical information has to do with Valentine's Day, which is right around the corner. For those of you who buy flowers to give either to guys or to gals, you'll often notice that with the flowers is included a small packet of powder. That powder is meant to be added to the liquid that the flowers are in, and that packet is to prevent the production or to inhibit the effects of ethylene. So just as in fruits, where damaging the fruit causes ethylene production, cutting the flowers also causes the production of ethylene. And I want to say, if you don't have that little packet, one of the things you can do is take an aspirin, not for yourself, take an aspirin, and put the aspirin in the water. And the aspirin also has effects which counter the damaging effects of the ethylene. Well, have a nice Valentine's Day, and I'll see you on Wednesday.